A patient arrives in the emergency department rigid, unresponsive, and locked in a generalized tonic-clonic seizure. The immediate assumption is often an epileptic focus, a cluster of neurons misfiring and triggering a cascading electrical storm. Framing electrolyte-induced seizures as a high-level network error is a mistake. The thalamus and the cerebral cortex are not deciding to misfire. They are victims of their environment. The true culprit is the extracellular fluid. When the concentration of specific ions in the fluid bathing the brain changes, the core biophysics of the cell membranes, the osmotic pressure, and the synaptic transmission mechanisms are physically altered. This graphic displays the 5 major electrolytes dictating cellular excitability, each a distinct mechanical parameter. Sodium acts as the spark. Potassium is the reset ion. Calcium serves as the threshold guard. Magnesium acts as the anti-overexcitation shield. And phosphate operates as the battery pack. This diagram illustrates the normal state of electrolyte homeostasis. Ions work in a precise equilibrium around every neuron, glial cell, and muscle fiber to maintain strict control over electrical impulses. To understand macroscopic clinical crises, why one imbalance causes flaccid paralysis and another causes violent tetany, we have to dismantle this microscopic cellular machine, one ion at a time. Sodium is the primary depolarizing ion for fast neuronal action potentials. A steep gradient from the outside to the inside of the cell provides the immense inward force needed to create the upstroke of an electrical signal. This creates a physiological paradox. When extracellular sodium levels plummet, the resulting seizures are not caused by a weaker inward electrical current. A lower sodium gradient actually makes generating that electrical spark harder, not easier. This section of the diagram outlines the major disorders of sodium, starting with hyponatremia. Dropping extracellular sodium lowers tonicity outside the cell, creating an osmotic gradient. To balance this, water aggressively shifts into the brain cells. The neurons and surrounding glial cells physically swell. This cerebral edema drastically compresses extracellular space. The physical crowding mechanically raises network excitability, triggering confusion and seizures. Hypernatremia operates in reverse. High extracellular sodium violently draws water out. The cell shrinks, placing immense physical stress on the neural network and disrupting signaling. Sodium disorders are a water problem first and an electricity problem second. Calcium is the classic molecular trigger for muscular tetany and seizures. Calcium performs 2 completely distinct jobs depending on its location. Inside the presynaptic terminal, its arrival triggers the fusion of vesicles and the release of neurotransmitters. But outside the neuron, it acts as a mechanical stabilizer. This schematic shows how shifts in membrane properties alter excitability. Extracellular calcium sits on the outside of the membrane, acting as a physical brake on voltage-gated sodium channels. It guards the electrical threshold required for the neuron to fire. In hypocalcemia, extracellular calcium drops. The molecular brake is removed. Without calcium stabilizing the membrane, the voltage threshold for an action potential physically drops closer to the resting state. Low calcium also increases activity in NALCN channels. NALCN stands for sodium leak channel non-selective, which provides a steady background inward current. This actively pushes the resting membrane potential upward, further closing the gap to the threshold. This graph illustrates the clinical result of hypocalcemia. The neurons require vastly less provocation to fire. The disinhibited sodium channels trigger rampant, runaway network excitability, resulting in generalized tetany. Conversely, hypercalcemia places too much calcium on the membrane. The neuron becomes overstabilized. The threshold is pushed so high that firing becomes incredibly difficult, inducing profound lethargy and weakness. Hypocalcemic seizures do not happen because more calcium enters the cell. They happen because the stabilizing guard goes missing from the outside. Magnesium serves as the necessary partner to calcium. It acts as a direct physiological antagonist to overexcitation across multiple tissues. Its primary mechanism of action in the brain is maintaining a voltage-dependent physical block on NMDA receptors. NMDA receptors are the major excitatory channels in the nervous system, activated by the neurotransmitter glutamate. This diagram shows what happens during hypomagnesemia. The protective magnesium plug is lost. Without it, the NMDA receptors stay open, entirely removing the restraint on excitatory glutamatergic signaling. Low magnesium frequently causes a secondary drop in extracellular calcium, compounding the excitability of the entire network. Stripping away the magnesium shield unleashes unmitigated excitatory drive. The clinical output is a severe escalation of tremors, arrhythmias, and seizures. Potassium shifts the focus away from explosive seizures and toward profound weakness. It governs the resting membrane potential and handles the repolarization that resets the cell after it fires. This chart displays resting potential changes. In hypokalemia, extracellular potassium drops. The concentration gradient pulling potassium out of the cell increases, which causes the membrane to hyperpolarize, becoming much more negative. The electrical threshold is now significantly farther away. The cell is vastly less likely to fire, presenting clinically as severe muscle cramps and flaccid weakness. This graph plots the two-phase danger of hyperkalemia. In phase 1, the outward gradient shrinks, causing a slight depolarization and a brief window of high initial excitability. In phase 2, sustained depolarization freezes sodium channels in an inactivated state. The cell cannot fire at all. Whether the gradient pushes the threshold out of reach or forces the channels into sustained inactivation, potassium disruptions ultimately exhaust the cell. The result is total muscle weakness and fatal cardiac conduction blocks. The final mechanism steps entirely away from channel gating and gradients to address the foundation of cellular economics, energy. Phosphate is the critical substrate for the synthesis of ATP. ATP, or adenosine triphosphate, is the primary molecule used by cells to store and transfer energy. This panel shows the cellular effect of hypophosphatemia. Severe depletion of phosphate leads to an immediate and direct cellular energy failure. Without ATP, the energy-dependent ion pumps that maintain the normal gradients seize up entirely. Cellular homeostasis is destroyed from the ground up. Without the cellular battery pack, the exact behavior of other channels becomes irrelevant. The biophysical system collapses, leading to deep encephalopathy, leakness, and respiratory failure. From a macroscopic clinical perspective, emergency departments rely on a rapid diagnostic framework to parse these completely distinct mechanisms. This summary table acts as a diagnostic matrix for interpreting electrolyte phenotypes based on the specific biophysics failing in the patient. If the patient presents with severe volume issues, specifically cerebral edema and subsequent confusion, the diagnostic logic points to the osmotic pressure of sodium. If the patient presents with unmitigated hyperexcitability, generalized tetany, or a sudden onset of seizures, the system is missing its threshold guards. This points directly to a lack of calcium or magnesium. If the dominant phenotype is profound weakness, lethargy, or a cardiac conduction block, the cell is exhausted. This indicates an altered resting potential driven by potassium, or a total energy failure driven by phosphate. True clinical mastery requires looking past the seizure or the paralysis. It requires visualizing the exact molecular mechanism failing at the cell wall. Electrolyte homeostasis is not a matter of keeping lab numbers neatly within a reference range. It is the strict, unforgiving biophysical tension required to produce every single thought, movement, and heartbeat.